Hi everyone, I'm Belinda, the RNA product manager of BGI Tech. I really appreciate that you joined our webinar and allow me to present our tips and advice for RNA sequencing. So for the next 1 hour, the first part is for wet lab tips introduced by me, and another part is for dry lab data analysis introduced by my colleague. My presentation will have 4 parts in total. The first part is an introduction about RNA. The second part is about the RNA sequencing procedure. This is the most detailed part. And the third part is highlighted features for RNA sequencing. And finally, I will summarize what you should consider when starting an RNA project. Before we start, please allow me to introduce BGI. BGI was founded in 1999, and BGI Tech Solutions was established in 2012, aiming to be an outstanding scientific solution provider for life science research community. And our service covers more than 100 countries and regions, and we have labs globally. We offers a comprehensive range of service and products as it present here, omics for all, including the genomics, transcriptomics, epigenomics, proteomics. This omics allows researchers, healthcare professions, biotech institute to have a better understanding about the complexity of life science. For omics research, it can be divided into several branches, such as the DNA sequencing, RNA sequencing, single-cell sequencing, and analysis platform. Today, I'm going to talk about the RNA sequencing part only. Let's move to our topic. Before talking about RNA sequencing, we need to know RNA first. Actually, RNA-seq is quite popular tools for biological research. You can see from the chart here, the RNA-seq related scientific publications increased year by year for the past 15 years. Then how does RNA generated and where are they from? The central dogma. From existing DNA to make new DNA is DNA replication. From DNA to make RNA is a transcription. And from RNA to make the protein is a translation. The RNA is here. The RNA transcripts are divided into 2 types, coding mRNA and non-coding RNA. The non-coding RNA divided into regulatory RNAs and housekeeping RNAs. With the different lengths of the regulatory RNA, the long coding RNA, which is normally above 100 nucleotide long, and the small RNAs are less than 50 nucleotide. This feature I show is a typical representation of the proportion of RNA component. For example, this is from the E. coli sample. The ribosomal RNA account for around 8% of the total RNA in the cell, while the tRNA account for 15% and the mRNA only account for less than 5%. As far as total RNA is concerned, the proportion of various RNAs may be different in different species, different tissues, different growth stages, different treatment. Let's learn more about the mRNA. For the mRNA, which is short for the messenger RNA, it is a single-strand molecule of RNA that corresponds to the genetic sequence of a gene and is read by a ribosome in the process of synthesizing a protein. In the eukaryote, a fully processed mRNA includes a 5' cap, 5' UTR, the coding region, and the 3' UTR and the poly-A tail. Actually, the alternative splicing is normal in the eukaryotes and may cause a single gene to be translated into multiple proteins. In prokaryotes, the mRNA structure is different from the eukaryotes. They don't have the 5' and cap and poly-A tail. In general, due to the lack of the poly-A tail and the cap structure, the prokaryotic mRNA are less stable than the eukaryotic ones and may degrade it very rapidly. Also, due to the lack of the poly-A tail, The RNA sequencing library processing is different. Non-encoding RNA are a type of RNA defined as being transcripts with lengths exceeding 200 nucleotides that are not translated into protein. Link RNA can be spliced, capped, and or polyadenylated and localized either to the nucleus or the cytoplasm of the cell. For the small RNAs are short RNAs, non-coding RNA molecules. It can regulate the gene expression. It includes the microRNA, siRNA, and piRNA. The most common sequenced RNAs are mRNA, long-encoding RNA, and the small RNA. Due to a different length and structure of those type of RNA, the procedure of RNA sequencing is different. So here's DNB-seq transcriptome sequencing and PacBio ISO-seq for mRNA, and DNB-seq link RNA sequencing product for long-encoding RNA. And actually, we can get both the mRNA information and the long-encoding RNA information from the sequencing data. And for the small RNA sequencing, the product is DNB-seq small RNA. Okay, let's see a little bit more about the mRNA sequencing. For mRNA sequencing, actually there are three main technologies based on different platforms. It can be characterized as short-read sequencing, long-read cDNA sequencing, and long-read direct RNA sequencing. In the short-read cDNA sequencing, as I show here, a significant proportion of reads map ambiguously when an exon is shared between the isoforms. And for the long-read cDNA method, it can generate full-length isoform reads that remove or substantially reduce these artifacts and improve the differential isoform expression analysis. Both these two methods rely on the cDNA conversion. For the direct RNA-seq, it can enable full-length isoform analysis, and also we can get the base modification information from the RNA directly. Then let's see why RNA sequencing. For RNA sequencing, different RNA type match with different RNA sequencing strategies, and transcriptome resequencing, it can reveal the precise quantity and structure of the mRNA. For RNA de novo, the transcripts can assembled and functionally annotated independently of the reference genome. And for ISO-seq, it can detect the full-length transcripts from the 5' and to the poly-A tail without assembly. And for non-coding RNA-seq, it's used for comprehensive research on the mechanism of the transcriptional regulations. For the application of RNA sequencing, it can reveal the growth and development, the stress resistance research for plants and animals in agricultural area, and for biomarker discovery and targeted drug development in the medical research. Also, it can be used for mechanism of the pathogenic regulation research. So in the previous part, I introduced the basic information about what is RNA, what type of RNAs, the RNA sequencing products, and the RNA sequencing application. Next part, I will bring you to our RNA project journey as a fresh user, from sample preparation to sample quality control, from library to sequencing and data analysis. The typical workflow for RNA sequencing starts from the sample preparation, then library construction and sequencing, data generate, and following data analysis. On each step, we will have the quality control to make sure that all the process goes right. Let's see the sample preparation. The very beginning of the RNA sequencing. Whenever we start an RNA sequencing project, we need to get the samples first. The starting material can be organs or tissues, cell lines, blood, FFP slides, and etc. When we have the tissue, we will go for RNA extraction first. Here, I would like to emphasize that the next part all I present is for bulk RNA sequencing, which is quite different from the single-cell RNA sequencing. Okay, when we have the total RNA extracted, the next step is RNA quality control. Why we need to do the RNA quality check. Because we need to gather information, the criteria about RNA, and evaluate if the total RNA is qualified or not for the following steps. The RNA quality evaluation is quite important. For RNA quality check, normally we need to check the RNA concentration, the purity, and the integrity. Here are listed 4 main methods for the RNA quality check. Some of the equipment may be easy to get from your lab. Some of the method requires specific equipment. For gel electrophoresis, which is very common in the biological lab, it can detect the purity and the integrity of the RNA. And for the nanodrop, it can detect the concentration and the purity. For the qubit, it can detect the concentration and the integrity of the RNA. And the Antigen bioanalyzer, it can detect them all. Next, I will show some case for that. For typical eukaryote species such as mouse, the total RNA gel electrophoresis result is like, here is a band for the 28S ribosomal RNA. This is a band of the 18S ribosomal RNA, the 5S ribosomal RNA. For high quality total RNA, very clear band of the ribosomal RNA here, and the brightness of the 28S is somewhere double the 18S. And here also are some cases for the low quality one, such as this one. You can see the genomic DNA contamination here. And for this sample, there's both protein and DNA contamination. And for this one, the band of the ribosomal RNA disappeared and you can only see smears. That means the total RNA is degraded badly. For the nanodrop, nanodrop using the optical density to reveal the concentration of the purity of the RNA. The OD260 over OD280 is to detect the presence of proteins and to a less extent the phenol contamination. And the OD260 over OD230 is specifically to detect the presence, estimate the degree of the salt removal, and the presence of organic contaminants. Then let's see the qubit. The qubit measurement system uses fluorescence technology to quantify the nuclear acids such as the RNA. The fluorescent dyes that selectively bind to the RNA molecules. These dyes only emit a fluorescent signal when they are bound to the targeted RNA, which allows for the specific detection and quantification of RNA without interference from the other molecules like DNA or proteins. They use a IQ score to represent the ratio of the structured large RNA molecules to small RNA molecules in the sample to evaluate the integrity of the RNA. Then the Antigen bioanalyzer is widely recognized as a gold standard for RNA quality check. There are many different types of the analyzer. Actually, they create the ring value, which is short for the RNA integrity number. It is a crucial metric for assessing the quality of RNA samples. The ring value counts from 0 to 10. The lower the score, the more degraded the RNA. This counting model is actually trained based on lots of data from mammalian animals, mainly from the human and mice. And some of the Antigen instruments use the ring value. Some of the equipment uses RQN value. Next, I will show you some cases. A typical Antigen testing result, the x-axis can be time or fragment size. The y-axis is the RFU value. The larger this value, the higher concentration of the sample. For eukaryote species, the high quality test result is shown like here. A very clear peak at the ribosomal RNA. This is a 28S, this is a 18S, and the line here is very smooth and flat to the baseline, means no RNA degraded fragment here. And it will show a very high, show the ring value with very high ring value, normally above 7. And here in picture B1, we can see another peak above 6,000 nucleotide long, indicate that this is genomic DNA contamination peak. And for the RNA degraded, the peaks of the ribosome RNA were disappeared, you can see from here. And we can see many small peaks in this region, and they are mainly the degraded ribosome RNAs. Next, I will show some of the testing results of spatial sample types. For the human tissue, such as the FFPE total RNA, because the treatment of the tissue, the total RNA were degraded, and the ribosome RNA peak disappeared, and we can see the small fragment in this region. And the rim value is 2, which indicate a really bad integrity of the total RNA. This is a very normal situation for FFPE samples, especially which stored for a very long time. For such as the sperm samples, plasma, and exosome RNAs normally contain small RNAs, degraded ribosome RNA or mRNA or other RNAs. So the peaks are normally in the region of less than 200 nucleotide, as we can see here. Let's see another interesting case from the insect. For insect, even with a very high ring value, which indicate a high quality RNA, but we can see that the 28S ribosome RNA peak disappeared. Why? It turns out that the heat denaturation step will break the large subunit ribosome 28S ribosome RNA into 2 fragments, the alpha unit and the beta unit, where the size are almost similar to the 18S. This will cause the fragment to be resolved at a similar position with the 18S in bioanalyzer profile. This phenomenon is explained in the paper, Why Does Insect RNA Looks Degraded? Let's see another demo from the plant leaf sample. The ribosome RNA type is different from animals. There are much more type of ribosome RNA peaks. Actually, the plant leaf samples, the RNA include the ribosome RNA from cytoplasm, mitochondria, and chloroplast with different types and lengths. Also, sometimes we will receive samples from eukaryote species, but we can see the typical prokaryote ribosome RNA peak in the profile. It turned out to be that the eukaryote samples that infected with bacteria. This is very common when we harvest the total RNA from the cell cultures in which were contaminated by prokaryotes. So the RNA quality check is important, and we can see from the previous case that different species, tissue types may have different results. It is really very complicated, but don't worry. For all the RNA samples will be quality check at BGI's lab. And we will show you the detailed evaluation result for your sample, level A, level B, or level C, and show you why it's level A, level B, or level C, and help you to know the sample quality and help you to decide the following step of your project. Then when the RNA quality is passed, then we go for library construction. I will introduce our procedure for RNA preparation for all our RNA products. For mRNA sequencing with DNB-seq platform, we start with the total RNA, and then we enrich the mRNA by combining the oligonucleotide bits with the poly-A tails of the mRNA. And then we go for fragmentation, PCR, and library QC. For different sample species and tissue type, there's different sample requirements. Also, we have protocols for difficult samples such as FP and low input RNA samples. For link RNA, because the long-encoding RNA may have or don't have a poly-A tails, so the poly-A selection is not suitable to get all the long-encoding RNAs. We will use a ribosomal RNA removal to enrich both the mRNA and long-encoding RNA at one shoot. This is the most common way for link RNA library construction. And that's the reason why for link RNA sequencing, the data contains both the mRNA and the long-encoding RNA. For small RNA library, The small RNA, they are really short, right? We were using the size selection by gel electrophoresis to enrich them during the library preparation. On the left is a procedure of the small RNA library preparation. Here's one thing that I need to mention that for fruit fly samples, the 2S ribosomal RNA has a similar size to the small RNA. So we need to eliminate the ribosomal RNA and only recycle the RNAs in this region. And we need to be very careful when we do the gel selection. Otherwise, we will see many unwanted ribosomal RNAs in the small RNA sequencing data. It is really wasting. And also we have a low input solutions for spatial sample extracted from the small RNA with starting material can be as low as only 5 nanogram. Then the PacBio ISO-seq. The ISO-seq is a full-length mRNA sequencing technology, also started with a total RNA, full-length cDNA synthesis, and PCR, and load on the PacBio sequencer. Actually, there are two types of ISO library. One is the non-size selected library, another is size selected. With a size selection in which it can help you to detect the longer ISO forms respectively. After the quality check, a library quality check, then we go for the sequencing part. The sequencing strategy and recommended data amount for each RNA sequencing type. 20 million per n reads is enough for mRNA sequencing, and 40 million reads is recommended for linker RNA sequencing. For the 20 million reads is enough for the small RNA sequencing. And for PacBio, since it's full-length mRNA, 5 to 10 million reads per sample can be enough. What's more, if you want to look deep into the really low abundance RNAs, more sequencing data will be suggested. Then go for the analysis part. We have standard pipelines and we generate web-based demo report for you to know the detailed parameters and analysis results. The next part, my colleague will introduce our RNA analysis system in details. So I just quickly go through it. So that's a basic procedure of our RNA sequencing from sample to analysis result. Next, I would like to introduce our technical highlights. The first one is a UMI technology applied in small RNA and ISO-seq. The UMI is short for unique molecular identifier. We know that during the PCR step, it will introduce a PCR bias in which will affect the quantification result. But with the UMI, it can increase the quantification accuracy by eliminating the PCR bias. How? Okay, let's see from this example. Here are 3 kinds of small RNA. RNA1 with 1 copy, small RNA2 with 2 copies, and small RNA3 with 3 copies. After the PCR, due to the PCR bias, we may see from the sequencing result that there are 3 copies of small RNA1 and 7 copies of small RNA2 and 5 copies of small RNA3. So can we say that the expression level for these 3 small RNA is 375 respectively? The answer is no. Just to look back to the original sample with the UMI here. It can eliminate the PCR bias and represent the original expression level of these 3 samples. That is 1 for small RNA1, 2 for small RNA2, and 3 for small RNA3. Okay, with the UMI technology, it can give you a more accurate gene quantification results with lower input, especially for exosome samples, and with for high reproducibility and repeatability. The second technology is the multi-throughput ISO-seq, as we know that the cDNA length is somewhere between 1 to 10 kilobase, but normally 2 to 3 kilobase for most of the human samples. But the PacBio sequencing reads can be as long as 80 kilobase. So each cDNA fragment were sequenced round and round for several times. Now, BGI have developed the technology to link 2 to 3 cDNAs together, and then we can generate more useful data in one single PacBio well. So we can largely increasing the detection of the transcript. Actually, this technology is developed by BGI, and we have the published the method on this article. And the third technology is poly-A sequencing. BGI developed the technology to reveal the full-length poly-A tails of the transcript. Here we can see from the right picture that the poly-A length varies from 1 to 300 nucleotides. But with PacBio's standard protocol, the poly-A length is our BGI's protocol is much longer than the standard protocol from the PacBio. With this technology, it can reveal the mechanism between the poly-A tails and the half-life of transcripts, gene expression level changes, and variable splicing events. Also, the method of the poly-A sequencing is also with the articles published. Okay. The fourth technology is a DocTom system. This system can simplify your data mining process without programming. For example, when you have BGI for transcriptome sequencing and data analysis, you will have all the charts and tables on DocTom system for basic results. What's more, you can adjust the sum of the criteria for narrow down the target genes or pathways, or take out the outlier samples and get a new table and new chart. There are more around 30 visualization tools on this platform. So for summary, when starting the RNA project, important tips are listed below. For your study design, we shall consider the scientific questions and considering your budget, and also about the number of the samples, how many biological replicates you should have, and your sample type and your sample quality, your RNA target and your analysis target. Based on your study design, when you talk to a scientific service provider, I recommend you to consider the checklist below. If they are experienced, do they have enough capacity and capability, or do they have a great quality assurance that finally can deliver the robust data and analysis results for your research? Actually, for RNA, we've been offering RNA sequencing since 2009, and we've been processed more than 1 million samples by now, and more than 2,000 articles were published using the DNB-seq RNA-seq from our clients globally. If you have any plan about starting an RNA project, or you have any inquiries about RNA sequencing, please feel free to talk to us. This is contact information. So that's all for my session. Please allow me to introduce my colleague for the next session.
Okay, thanks for Yao's introduction. And now we know that the RNA-seq are widely applied almost in any research of investigating biological mechanisms. Thus, the analysis of RNA-seq data becomes a huge need. And actually, since RNA-seq technology has been developed for a long time, there are mature pipelines to analyze the data. And basically, it can be divided into 4 modules. Firstly, the data processing. Low-quality data will be removed, and the high-quality data will be aligned to the genome and gene set to check if there are exogenous contaminations. And this is the fundamental step to get guaranteed results. Next, the gene expression can be determined. Thus, we can identify differentially expressed genes, which is the most important result in RNA-seq analysis. And besides, variations in RNA level, including new transcripts discovery, alternative splices, SNPs, and indels, can also be checked. And after getting the basic expression information, multiple advanced analysis can be done. For example, the enrichment analysis of DEGs to identify targeted pathways or gene sets, the PPI interactions between genes, and multi-omics analysis, etc. And although the analysis of RNA-seq data is almost the easiest one in NGS data analysis, there are still many algorithms that need to be learned. Not everyone knows how to use R or Python, like me. And if you seek for the help of core facilities or other partners, the turnaround time may be long. Or if you need other analysis out of the standard pipelines, repeated communications may be needed. And worse still, core facilities may not accept personalized requests. And that's why we did cite Dr. Tom system years ago. We are aiming to help researchers to interpret the data and visualize them. Even those who are not bioinformaticians can perform the RNA-seq analysis easily and conveniently. And right now, we have more than 20,000 users all over the world. More than 350 papers were published, in which they've cited Dr. Tom. And we've built powerful databases of more than 100 species on Dr. Tom, in which the information from public databases, such as KGG, GO, and BGI in-house data were collected. And for data visualization, more than 20 tools can be used. Users can select data as they want and adjust the color scheme to generate figures that can be published directly. And since gene transcripts and proteins information are all collected in Dr. Tom, it's possible for users to perform multi-omics analysis. With the amazing design of table chart interaction, it's easy for users to perform unlimited and deeper data mining. And moreover, Dr. Tom accepts gene expression data from any sequencers or service providers. Even if you did not sequence the samples in BGI, users are able to upload their own data and use Dr. Tom to analyze them. So I would like to show you how amazing Dr. Tom is right away. But before that, let's take a look at the paper that used Dr. Tom to analyze the RNA-seq data to see how it can be applied in transcriptome data interpretation. So this paper focused on the role that the endocannabinoid system plays in the endometriosis pathogenesis and immune response. And CNR1 and CNR2 are important receptors in the endocannabinoid system, which regulated various physiological processes, including immune response, pain perception, etc. And in the RNA-seq experiment of this paper, decidualized and undecidualized endometriosis lesion mouse were tested. And in each type, there were 3 groups: CNR1 knockdown, CNR2 knockdown, and wild-type mouse models with endometriosis. And in RNA-seq data analysis part, a total of 1,106,639 DEGs were identified in both undecidualized and decidualized lesions of CNR1 knockdown and CNR2 knockdown mice, respectively, compared to the wild-type controls, as summarized in the bar chart. And taking the decidualized type as an example, the KGG enrichment analysis were performed. And in decidualized lesion from CNR1 knockdown sample, notably, the cell adhesion molecules pathway was prominently affected. indicating a potential role for CNR1 in mediating cell-cell interactions and tissue remodeling processes. In the visualized lesion from the CNR2 knockdown mice, the analysis highlighted distinct pathways affected in the context of inflammation and endometriosis, including the cytokine receptor interactions pathway, pointing to the involvement of CNR2 in immune responses and inflammatory processes associated with endometriosis. And a total of 59 key genes in endocannabinoid signaling were checked. As shown in the Venn graph, only 3 and 2 DGs out of the 59 genes were identified in the visualized lesions of CNR1 knockdown and CNR2 knockdown, respectively. And to see what functions these 59 genes may have, the comprehensive gene oncology classification analysis on the 59 identified endocannabinoid signaling genes unveiled their multifaceted roles in reproductive functions, immune system regulation, and cellular processes. These figures and data interpretations shown here are all pictured by Dr. Tom. And in fact, it's quite easy to perform DGs analysis, use DGs to do enrichment analysis, draw a bar chart to show geo-annotations on Dr. Tom. And let's switch to Dr. Tom and see how it works. So this is the demo project of transcriptome in Dr. Tom. And in BGI, the analysis result will be provided on Dr. Tom, and we divide them into 4 parts: basic information, expression, gene annotation, and variation. And when we hover on each module, you can check the specific results. For example, the DG bar chart here in the expression module, the enrichment bubble plot, and the classification bar chart here in the annotation module. And BGI provide the results not only in a clear structure, but also a delicate and practical pattern. And the plot can be directly downloaded and used in the publication. And if you prefer other visualization tools, you can download the plotting data from Dr. Tom directly at any time. And beyond the visualization of the result, Dr. Tom also makes the adjustment of the plot easy. By clicking the color box, you can change the color. And by double-clicking the title, you can also change the title here. And if for DG analysis, if you want to adjust the filtering parameters, click the adjustment here and change it immediately. So this is the adjustment on the original results that BGI analyzed and provided. And what about customized analysis? And as you can see, there is a green button on the bottom right of each table. Click that, you will see like around 20 tools can be used freely, including commonly used enrichment, GSEA, heat map, etc. And for example, if you want to set a totally new comparison group that is not included in the previous analysis plan, the difference analysis tools can help you. Here you can select the analysis software, set the customized groups here. For example, I will randomly select some samples. And you can also set the filtering parameters here. And click OK to submit the analysis task. And you can check all the analysis in My Analysis. And usually we will need a few minutes to complete the analysis. And you can check the analysis result here. So this is the task I've done before. So we will check these differentially analysis results. And the most amazing design of Dr. Tom, I think, is the interaction between plot and data table. For example, when we hover on the bar, we know that there are 5 upregulated genes, but what exactly these 5 genes are? And when I click on this bar, the right side table will change correspondingly to show the details of these 5 genes. And this table shows the gene ID, gene symbol, type, the expression, and log2 change, the p-value, and q-value by default. And if you want, you can add information by this extend button to extend this table. For example, I can add the annotation of these genes in KGG pathway. Click OK, you will see that the annotation will be added to this table, and you can also download this table. Now we've got these 5 genes, and somehow I want to display the expression of them in all the samples in the heat map. So again, I can directly open the toolbox, find the heat map tools, and set the parameters here, and again, submit the task. And we can also check the heat map I've just done. And on the heat map, we can also select the genes we are interested in and use other tools to visualize them. So the design of interaction between plot and tables, in addition of abundant tools, allows us to interpret the RNA-seq data step by step, deeper and deeper. So due to the limitation of time, I cannot show you all the detailed functions of Dr. Tom. And if you are interested in Dr. Tom, you are welcome to register in a free account on our website and try the demos. So that's what I want to share today. Thank you for joining us.